Hello everyone, I am Vinicius Thomazi. I'll be presenting our work that has been developed at the Stem Cell Laboratory at Rio Grande do Sul Federal University, and it's titled Assessment of Gelatin Methacrylate Cross-Linked with Natural Photoinitiators and Visible Light for Neural Applications. At the Stem Cell Laboratory, we work with regenerative medicine, and we aim to repair and regenerate different tissues. And in particularly in this work, we are working with neural tissue, and it is challenging because neural tissue is very soft. And we develop biomaterials, and they have to match the physical chemical characteristics of the target tissue. So it's a challenge to create a very soft and yet resistant material. One of the most versatile materials that we work with is gelatin. Gelatin is broken down to collagen, and it's biocompatible, and we may tweak the mechanical characteristics of it by changing the concentration. But gelatin melts at physiological temperatures at 37 degrees Celsius. It melts. So one of the strategies is to functionalize it with methacrylate groups and create GELMA. And then this GELMA may be cross-linked, so the molecules cross-link with each other and create a much more resistant material than the original gelatin. And we do that by using photoinitiators. There are molecules that are stimulated by light, and they produce free radicals, and these free radicals react with the methacrylate groups in GELMA, and we finally have our cross-linked construct. So we may see in figure 1 that on the top we had two pieces of gelatin, and at the bottom in 1b we see that the left one melted after being exposed to 37 degrees Celsius, and the right one is GELMA after cross-linking, so it kept its form and it's resistant to temperature. So we produced the GELMA in our laboratory, and it's a laborious process. It takes several days because you have to dialyze it extensively because the initial chemical reaction produces toxic waste. But then we tested it with the neuroblastoma cell line, SHSY5Y, because our end goal is to use it for neural applications. And we use photoinitiators, and this is key to our work, that were all natural. We used riboflavin, which is vitamin B2, and we used arginine, which is a naturally occurring amino acid, because most photoinitiators are synthetic, and more than that, a lot of times they need light that's very energetic on the ultraviolet spectrum, and this is damaging to cells and molecules. So a molecule that you might want to add to your biomaterial may be damaged because of the ultraviolet. So riboflavin and arginine cross-link with blue light. So they produce the free radicals in the blue light spectrum, which is visible light, and we hoped would be less damaging to cells. So we tested it. We had two groups of GELMA, one pre-cross-linked and other the cells were added before cross-linking, so they took the full blast of the 15 minutes of blue light, and then we had controls exposed to light or not. And our results showed that the cell viability as assessed by the WSTH assay was very similar between the groups, and the two GELMA groups had slightly higher cell viability, only the not exposed to light was actually significantly higher than the controls. And we may see in figure 2 the aspect of the cells in 2A in GELMA, because it is a 3D culture, it becomes more round. And in 2B, we can see the natural shape of the cells that resemble neurons much more. So it's a very exciting material and a very promising technique to cross-link it at blue light and at 10%, which is a low concentration that could match the neural tissue. So we'll keep developing it. We are doing it right now, and I thank you all for your attention and your interest in our work. Thank you very much.